Meanwhile, researchers say type O blood appears to offer people some protection against the coronavirus. Two studies published in Blood Advances suggest people with type O may have a lower risk of coronavirus infection than those who have other blood types.
If you're fascinated by the edge of discovery, where science meets history and biology rewrites everything we thought we knew, you're in the right place. This channel explores mind-blowing breakthroughs, hidden truths, and science's deepest puzzles, all backed by the latest research and findings. We're not just scratching the surface, we're going deep into the why and how of the world's greatest mysteries. Whether it's ancient genetics, biomedical breakthroughs, or the evolutionary secrets locked in your DNA, our mission is to deliver the facts, decode the science, and share the human story behind each breakthrough. Today's deep dive is one that touches every human being's blood type, and not just any type. The elusive, fascinating, and life-saving type O blood. What makes it so special? And why is it particularly mysterious in the Americas? In 2025, scientists finally cracked the code. Let's dive in. Type O blood is more than a classification. It's a key that unlocks mysteries in emergency medicine, genetics, and human history. Its biological value is enormous, especially when seconds count. In trauma situations, doctors don't always have time to type a patient's blood. That's where type O negative becomes critical. It can be transfused into anyone, regardless of their blood type, making it the default option when there's no room for error. This is why every emergency room, military base, and ambulance aims to stock it. Type O positive also plays a crucial role. It's the most common blood type worldwide, especially in places like the Americas and parts of Europe. While not universally accepted like O negative, it can be transfused into any patient with a positive Rh factor, making it incredibly useful in large-scale emergencies where blood demand spikes. But what really turns heads is type O's strange global pattern. Scientists noticed something unusual when mapping blood types across continents. In certain Native American tribes, especially in South and Central America, virtually 100% of individuals had type O blood. That's not just statistically rare, it's biologically astonishing. In evolutionary terms, genetic diversity is the norm. Over time, interbreeding and migration tend to introduce variation. So even in relatively isolated populations, we'd expect to find a mix of A, B, and O alleles. But with some indigenous American groups, those other types are practically non-existent. This led scientists to wonder, how could such a narrow genetic trait persist in entire populations? For decades, researchers theorized, some believed it was a founder effect, that the first small groups to migrate into the Americas from Siberia during the Ice Age happened to carry only the O allele. Others pointed to natural selection, suggesting type O may have offered a survival advantage in environments plagued by specific pathogens. Then, in 2025, a breakthrough arrived. Genomic studies comparing ancient DNA from mummified remains, ice-preserved humans, and early American settlers revealed a strong pattern. The people who first crossed the Bering Land Bridge roughly 15,000 years ago came from a genetically narrow group. These early humans likely underwent a genetic bottleneck, a period when their numbers were so low that only a limited gene pool survived. And in that pool, the O allele dominated. What's more, it turns out that this allele conferred a modest advantage in terms of resistance to certain infectious diseases, such as malaria and tuberculosis. While not completely immune, type O carriers were slightly more likely to survive outbreaks, allowing their bloodline to flourish while others diminished. Over generations, this created a sort of genetic echo. Type O blood embedded deeply into the fabric of American indigenous DNA. Even today, that ancient fingerprint persists. Across Latin America, type O remains disproportionately high, even in urban centers with diverse populations. It's a lingering clue from humanity's ancient migration story, one told not in bones or artifacts, but in the living tissue of blood. Understanding type O's role isn't just an academic pursuit. In a world increasingly reliant on blood transfusions for surgeries, childbirth, and emergencies, knowing where and how this blood type is distributed helps global health systems plan better. It shapes blood donation campaigns, informs medical research, and even offers insights into how our ancestors adapted to a changing world. So, the next time you see a call for blood donations, especially for type O, remember, you're not just giving blood. You're continuing a legacy that has flowed through humanity for thousands of years, a lifeline connecting past, present, and future. The founder effect has long been a cornerstone theory for explaining the unusual dominance of type O blood among indigenous peoples of the Americas. According to this idea, when a small group of early humans migrated from Siberia into North America across the Bering Land Bridge around 15,000 to 20,000 years ago, they carried with them only a limited set of genetic traits. If this migrating population was largely composed of individuals with type O blood, then, through generations of reproduction within an isolated gene pool, the O allele could have become predominant by default. This kind of genetic bottleneck isn't unusual in evolutionary history. But as researchers dug into this theory, it started to show cracks. While the founder effect explains how a single trait could become common in a small, closed population, it doesn't account for the near-total absence of the A and B alleles. Over the span of thousands of years, you would expect genetic mutations to reintroduce these alleles. Even modest interbreeding with other human groups would normally result in more blood type diversity. But in many Native American tribes, especially those isolated in the Amazon or high Andes, A and B types remain vanishingly rare, sometimes entirely absent. So, if not just genetic drift, then what? This opened the door to another possibility: natural selection through environmental adaptation. Could the environment itself have favored individuals with type O blood? Researchers began looking at specific traits tied to the O allele. One of the most compelling lines of investigation centered on disease resistance. Type O individuals, it turns out, tend to fare better against certain diseases. They're less likely to suffer from severe malaria, for instance, a disease that, while not historically widespread in North America, may have been a factor in parts of Central and South America. Even a slight survival advantage in these regions would, over centuries, lead to more type O individuals passing on their genes. Another fascinating angle came from studying digestion and metabolism. People with type O blood typically produce more stomach acid and possess a robust gut biome optimized for protein digestion. This would have been advantageous in early human diets that relied heavily on hunted meat and gathered food with fewer carbohydrates and grains. In environments where agriculture hadn't yet taken root, this digestive edge could have significantly improved survival, nutrient absorption, and resistance to pathogens in food. Still, these remained educated guesses until 2025. That year, an international team of geneticists and anthropologists conducted an unprecedented genomic study. By analyzing both modern and ancient DNA samples across the Americas, they were able to construct a high-resolution map of blood type distribution over time. What they discovered was groundbreaking. The persistence of type O wasn't just due to the founder effect. It had been actively preserved by selective pressure. Their data showed that individuals with the O allele had marginally higher survival and reproduction rates in certain environmental contexts, especially in regions with high exposure to zoonotic pathogens, where immunity and digestive efficiency played key roles. Over time, natural selection reinforced the dominance of the O allele in these populations, effectively crowding out A and B. In other words, evolution had a hand in preserving what the founder effect had started. The combination of a narrow genetic starting point and selective advantages turned type O into not just a common trait, but a defining one for entire populations. Today, this discovery reshapes how we understand human migration, adaptation, and survival. Blood, once thought to be a simple hereditary marker, now reads like a storybook, one that tells of isolation, resilience, and a deep biological memory of the environments that shaped us. In 2025, a groundbreaking genetic study redefined what we thought we knew about human blood types. Conducted jointly by teams from Harvard University, UNAM in Mexico, and the Max Planck Institute in Germany, the project leveraged next-generation sequencing to analyze thousands of DNA samples from indigenous populations across the Americas. What they found was astonishing. A previously undiscovered suballele of the type O gene, now called O', was present almost exclusively in Native American genomes. This wasn't just another variant. O showed unusual genetic stability, meaning it had resisted mutation for thousands of years. Compared to other blood type alleles, which tend to evolve and shift over time, O had remained virtually unchanged since its earliest appearances. What shocked researchers even more was its apparent age. O wasn't just another version of O, it might actually be the oldest of all blood types. This discovery upended decades of medical and evolutionary thinking. For years, the dominant theory held that type O was a stripped-down, simplified mutation of the more complex A and B types. But with O predating them all, a radical new hypothesis emerged. Type O may have been the original human blood type, and A and B might actually be the mutations, adaptations that developed later in response to environmental pressures and migration patterns. This reframing made the global dominance of type O in certain populations, particularly in indigenous American groups, make a lot more sense. Rather than being a leftover trait or a genetic accident, type O appeared to be the blueprint, the foundational format from which others deviated. Beyond the genetics, the research team also explored the social dynamics that might have helped perpetuate type O in early human societies. Even though ancient tribes had no concept of blood transfusions, blood was widely regarded as a symbol of life, power, and connection. Many indigenous cultures held spiritual beliefs linking blood to strength, ancestry, and divine favor. This could have influenced social perceptions of health and vitality, key factors in mate selection. Some anthropologists now suggest that type O individuals may have been unconsciously preferred as partners, especially if they exhibited greater resistance to disease or showed signs of physical vitality. In tightly knit communities where survival depended on resilience, even small advantages could tip the scale. Over time, if these individuals had more children, the frequency of the O allele would naturally increase without any formal understanding of genetics. Additionally, when epidemics struck, whether from native pathogens or later from European-introduced diseases, communities with a higher proportion of type O individuals may have had slightly better survival rates. While no blood type offers complete immunity, the protective features associated with O, such as a reduced risk of certain blood clotting disorders or inflammatory responses, could have had an impact. These small biological advantages, compounded over generations, might explain the incredibly high concentration of type O blood in these populations today. The 2025 findings didn't just clarify the biological mystery. They opened up a new lens through which to view the intersection of genetics, culture, and evolution. It revealed that our blood types are not just medical data points, but living records of migration, adaptation, and survival. Type O's dominance among indigenous Americans is no longer seen as an evolutionary accident. It is increasingly viewed as evidence of a deeply rooted genetic lineage, possibly the original human standard from which all others diverged. In this light, type O isn't just important for modern medicine. It's a legacy, etched into DNA, telling the story of who we were and how we made it this far. While type O blood has long been celebrated for its versatility in transfusions and its potential resistance to certain diseases, this evolutionary advantage doesn't come without trade-offs. Like most traits shaped by natural selection, there's a cost to the benefits. One of the clearest examples is the link between type O blood and peptic ulcers. Studies have shown that individuals with type O blood are significantly more susceptible to Helicobacter pylori, the bacterium that causes stomach ulcers. This bacteria binds more easily to the stomach lining of O-type individuals, increasing their risk of chronic gastritis and ulcer formation. It's a sharp contrast to the usual narrative of O-type as the strongest or most resilient blood group. In addition to digestive issues, thyroid dysfunctions appear more frequently in people with type O blood. Conditions such as hypothyroidism and Hashimoto's thyroiditis have shown statistically higher incidence rates in this group, possibly due to underlying immunological factors tied to their unique antibody profile. That brings us to another issue: autoimmune complications. Because O-type blood lacks A and B antigens, its immune system tends to be more aggressive in identifying and reacting to what it sees as foreign. While this can be beneficial when it comes to fighting infections, it can backfire. The very antibodies that make type O blood safe for donation can, in some individuals, contribute to overactive immune responses, increasing the risk of autoimmune diseases. Another lesser-known downside is related to blood clotting. Type O individuals often have lower levels of von Willebrand factor and factor VIII, both crucial for effective clot formation. This means they may experience longer bleeding times, making injuries or surgeries slightly riskier. While not severe in most cases, this subtle clotting deficiency can pose a concern in trauma care. So the question is, if these risks exist, why didn't natural selection phase out or reduce the frequency of the O allele over time? Why would a trait that increases susceptibility to ulcers or autoimmune disorders continue to dominate entire populations? Here's where the 2025 discovery of the O' suballele becomes pivotal. Researchers found that O, which is prevalent in many Native American populations, exhibits greater immune balance than previously known O alleles. Unlike regular O-type blood, O appears to avoid triggering excessive immune responses. Its antibody activity is strong enough to defend against pathogens, but seems less likely to lead to autoimmunity. Additionally, early lab studies suggest that O may include slight structural modifications to clotting factors that offset bleeding risks seen in traditional O types. This makes O a kind of evolutionary refinement, a hyperstable version of type O preserved and honed in genetically isolated populations for thousands of years. It likely helped these communities maintain the advantages of type O without fully bearing its costs. It also explains why the O gene pool remained so dominant in spite of its risks. But the implications of this discovery don't stop with anthropology or evolutionary biology. Medical protocols around the world are now beginning to change. Blood donation centers in several countries have started screening for the O variant as a potentially safer and more efficient universal donor line. Because of its balanced immune profile, O could reduce complications in emergency transfusions or organ transplants. There's growing interest in studying how the O protein markers interact with immune systems across different blood types, with the goal of engineering truly universal synthetic blood, something that has long been the holy grail of transfusion medicine. Further down the line, gene therapies could be developed using O as a model, potentially helping people with autoimmune disorders or clotting deficiencies by mimicking its unique genetic profile. There's also promise in transplant medicine, where O traits might reduce organ rejection rates. In essence, the 2025 genetic breakthrough didn't just answer an ancient mystery, it launched a new era in modern medicine. What began as a quest to understand a blood type anomaly may soon revolutionize how we treat disease, manage immunity, and save lives in critical moments. The story of Type O isn't just a medical marvel. It's a window into human history. Blood type maps closely with human migration, giving clues about ancient travel routes, genetic bottlenecks, and intertribal mixing. The widespread dominance of Type O in the Americas may be the best genetic breadcrumb left behind by the first peoples to cross into the New World. And now, thanks to the 2020 we know that their blood tells a story of adaptation, survival, and identity that we are only beginning to understand. What do you think about this game-changing discovery? Could it lead to synthetic universal blood? Does this mean our understanding of human evolution needs to change? Let us know what you think in the comments below. We read every single one. If you found this video fascinating, smash that like button, hit subscribe, and don't forget to turn on notifications so you never miss our latest scientific deep dives. This is more than science. It's the story of us. And there's so much more to uncover. Thanks for watching, and we'll see you in the next one.